Okay, so welcome to this lecture on genetic engineering and biotechnology. So I know everyone gets really scared when the word genetic or genetics is mentioned, but this is all capable of being rather straightforward, especially if you connect it to things that you already know or have covered in lab and previous lectures. So the first thing I want you to know is what it means when you hear the term recombinant DNA, okay, or recombinant DNA technology. So basically what you're doing with recombinant DNA technology is you are copying, cloning, or manipulating DNA, because recombinant DNA usually refers to DNA that has two different pieces of DNA put together. Okay, so recombinant DNA, think of it as a hybrid of different DNA pieces. Okay, so it has new genes that have been introduced to it. Okay, now when you do recombinant DNA work, meaning genetic engineering, you're going to use two key tools. The first one is restriction enzymes. Those you usually hear called nucleases. Okay, so make sure you write down the word nucleases to describe enzymes that will cut DNA. Okay, and these basically they're highly specific and will only cut certain sequences. And if that particular sequence is only found in the DNA sequence once, okay, you get one cut. Found 3, 4, 5 times even, the enzyme will cut every single time that it encounters that particular sequence. Okay? Now, in addition to using these nucleases, you also will use a cloning vector. So you've used those in lab classes while at Georgian Court. Some of them may have been P-Blue or the puck one used in the ligation and transformation kit. Okay, cloning vector is basically a fancy way of saying a plasmid. Okay, so you see the plasmid here, round piece of DNA. Now remember, plasmids are usually these circular pieces of DNA. They're extra chromosomal, meaning that they are beyond the life function chromosomes, and most importantly, these cloning vectors can give bacteria an advantage. And make sure you always know what advantage is it that a plasmid or cloning vector can give to bacteria? Antibiotic resistance. Okay, make sure you write down that cloning vectors can give bacteria antibiotic resistance. So when you're going to use recombinant DNA technology, or basically genetic engineering, a lot of times we call it molecular cloning. And what you have to do is you cut a target gene out of a genome, and then you cut a vector with the same nuclease, the same enzyme. Okay? And once you do that, those two pieces of DNA, your target gene that you cut out of a genome from either human or yeast or bacteria, that target gene that you cut out of a genome now has the same sticky ends as the vector that you just cut open with the same enzyme. So now what you're able to do, as you see in this figure on the slide, is you can ligate them together. Okay, you take that gene that you cut out of a big genome, and you take that vector that you cut open in just one spot, and you glue together those pieces. Once you have that DNA ligated, what can you then do? Well, now you could do transformation. And what's transformation? That's basically getting bacteria to take in that new plasmid, that vector, that has your target gene in it. And in the example on this slide, the picture, the target gene that was used is GFP, or green fluorescent protein. And so now that bacteria that has the plasmid with the GFP that you put into it, well, now they can glow green in the presence of UV light. So that's what I said in lab courses when you got to do transformations. It's a very powerful tool. You can give bacteria new genes, which gives them new abilities. They can produce completely new enzymes or proteins that they never had the ability to produce before. And like I said before, remember that one of those things that you can give them is antibiotic resistance, okay, which is very beneficial to the bacteria, and it's also useful in your research as well to help you find the successful transformants later. Now, this is something that you may have seen or remembered from lab, okay? When we talk about cloning vectors, there are 4 main qualities that a cloning vector or plasmid should have in order to be valuable for genetic engineering or transformation, okay? The first one is that the plasmid should be fairly small. And when you ask yourself, why should this plasmid be fairly small? Well, think about what you want to do with it. You want that DNA ring to get into bacteria. And remember, those bacteria have teeny tiny little pores on the outside of their outer membranes. So if that plasmid is nice and small, that increases your chance of getting the bacteria to get that DNA inside of it. Okay, so for the first why should the plasmid be fairly small? To help it get into the bacteria. The next bullet point we have is that the plasmid should have an origin of replication. Why is that? Well, it's in the name. You want that DNA to be copied and maintained in the bacteria once the DNA has gotten in. Okay, so the origin of replication allows the bacteria to copy and maintain your plasmid genes. Okay, then we say that the cloning vector should have at least one selectable marker. Okay, and think of the labs that you've done with transformations. What are some selectable markers that we tend to use? Well, one I've mentioned repeatedly already in the few minutes of this lecture, which is antibiotic resistance. The other is a color change, okay? So if at the end of this experiment, after incubating, your bacteria now can grow in the presence of something like ampicillin, or if it suddenly turns blue when it grows in the presence of something like Excal, well then you know those bacteria successfully took in your plasmid, okay? So that's why selectable markers are so valuable, is that they allow you to see which of your bacteria successfully took in the plasmid, okay? Which are the successful transformants. The last of the bullet points is the word that students always hate. They always forget how to say it. That's endonucleases. And endonucleases, what they do is they cut open DNA. So if you remember when we talk about molecular cloning, we said that you want to be able to cut this plasmid open, put new genes into it. Okay, so that's the why of restriction sites or endonuclease sites, is you need to be able to manipulate that plasmid. Okay, you need to be able to cut it open. And usually we call these MCS, which stands for multiple cloning sites. Okay, multiple cloning sites, because these are sites on the plasmid that allow for molecular cloning. They allow you to cut open the plasmid and then insert new genes. Okay, so I'm going to have a brief recap on the next slide, just so you get time to write down what we just said. Okay, so here is the recap slide. You can pause the video and take down the notes in case you missed anything I said in the last slide. Okay, so now when we talk about plasmids, A lot of times you'll hear them called multiple copy or multi-copy plasmid vectors. Basically that means, so I'm going to bring all the bullet points out here. Basically that means that these plasmids, especially if they have their proper origin of replication, they can make multiple copies of themselves within the bacteria so that the bacteria can then have high levels of them, okay, which is very valuable to us as researchers because basically the bacteria can become little factories for us, okay, because if they have multi-copy plasmids, they can make many, many copies of the plasmid and then express those new genes at high levels for us. Okay, now one of the most commonly used ones today, it came from what we call the CoL-E1 plasmid that E. coli naturally had. And what researchers did was they took this original, so if you look on the slide here, they took this original plasmid that they found in E. coli and they removed what was called the colicin production genes. And the reason they removed that is colicin is a bacteriocin. And what that means is it's a toxin that kills some bacteria. Okay, and you may ask yourself, well, why would a bacteria produce a toxin that kills bacteria? Well, it allows it to reduce competition. So especially if they kill off the more similar kind of slightly related bacteria, they get to hoard or have more of the resources they need. Okay, but you don't want that in your plasmid because then it would kill off your experiment. So researchers removed the colicin gene, and then they added what's called the beta-lactamase gene. Okay, they added antibiotic resistance. And why would they want to add the beta-lactamase gene? Well, now if you use this plasmid in research, if it gets into the bacteria, then you'll know because that bacteria will now be able to survive on ampicillin plates. Okay, so you've done this kind of transformation in multiple of your classes, most likely by now. Okay, so keep in mind when you think about plasmid vectors, remember that the original one was from E. coli, and they had to remove the colicin gene and add in the beta-lactamase gene. Okay, and understand why they wanted that beta-lactamase gene in there. So now we get to inserting genes into vectors, which we've, basically been talking about so far, but I want to kind of give you some general rules about doing this. So what you want to do in molecular cloning is you want to cut that plasmid and then cut the gene of interest with the same restriction enzyme. Okay, because we remember we said you're getting that gene of interest from another cell's genome. So you have to cut out that gene of interest and you want to use the same restriction enzyme so that it will have sticky ends that properly ligate to the vector that you cut open. Okay? Now, when you're going to do this, there's certain rules you have to keep in mind. The first one is when you cut, you should only cut the vector once. Okay, you want to open up the vector so that you can glue in the new gene. But what would happen if you cut the vector more than once? You didn't open it up now. You completely chopped it up and it becomes useless at that point. Okay, so you want to make sure you only cut the vector once. You also want to make sure you're not cutting into any genes that are necessary for plasmid stability or survival. If you cut an important gene, then you might end up killing the bacteria when you try to put that plasmid in, or the plasmid won't work. Basically, to help avoid problems, a lot of the vectors of the plasmids have been modified So they contain what's called a polylinker or the multiple cloning site we just mentioned a minute ago. That way you have a lot of different options, okay? You have multiple sites to choose from so that you're most likely not, kind of running into the problems we just mentioned. Now, once you have cut the vector and the gene of interest, the next thing you have to do is use DNA ligase to join the ends. So make sure you remember, if you're going to do genetic engineering, you have to cut the plasmid and you cut the gene of interest with the same enzyme. You only cut that plasmid once, and then you use DNA ligase, which is like glue, to stick together the gene of interest and the vector. Okay, so now you should have a nice full plasmid with an extra gene in it. This slide here is to show you what I meant by the polylinker and by the process that we just talked about. Okay, so it helps you visualize. Now, keep in mind, not all plasmids will have that insert or the polylinker, and ligation is not always perfect. Okay, when you do this process, not every vector is going to get cut open. Not every vector is going to get the intended gene, because remember, in lab we had mentioned that when you do this type of experiment, when you cut that vector open and then you put it with these DNA pieces that you want to stick into there, well, look at these. These guys also want to stick back to each other. They match. So a lot of times when you do the experiment, a lot of your plasmids will actually religate on each other and not have the insert. So basically, you need to make sure that there are ways for you to detect which plasmid successfully got cut open and actually took in the gene of interest, okay, which you see here with the plasmid and the gene of interest, rather than just sticking to themselves. So now on this slide, we're going to start going into some ways that you can detect whether your plasmid successfully took in the gene of interest. Now keep in mind, this is different from the selectable markers we talked about earlier. Things like color detection and antibiotic resistance, that was just to show if your plasmid has made it into the bacteria. And that doesn't tell you whether or not your gene made it into the plasmid, because remember, some of the plasmids will just stick to themselves, okay, rather than taking in the gene. So to try and determine whether your plasmid has successfully taken in the gene, you can do a few things. One thing that you can do is you can isolate from the bacteria, pull out the plasmid DNA, and you cut it with the same enzymes that you used in cloning. And think about it. If you look at this picture on the bottom of the slide, if you isolate plasmid DNA from the bacteria and you cut it with the enzymes from cloning, if that plasmid only religated to itself, which is this guy here, then when you cut it with that enzyme, it'll only cut once, and you'll see one linear strip of DNA. If, however, it took in the insert successfully, when you now cut it with the enzyme, look, that's 2 cut sites, right? So now when you run this on a gel, you'll see 2 pieces of DNA instead of 1 linear piece. Okay? If you have trouble visualizing that, just send me a remind message and I'll go over it in a little more detail. Okay, so what we just covered here, if you want to detect an insert in the plasmid, one way is to just simply extract the plasmid DNA and cut it with the same enzyme. If you see 1 band of DNA, then it is linear. It did not take in the insert. If you see 2 bands of DNA, then it did take in the insert. It successfully took in the insert. Another thing that you can do is insertional inactivation. which is what you did in laboratory if you perform the blue and white ligation transformation technique. And so we're going to go over that one in more detail in the coming slides. And then you have the option to use reporter genes, which kind of a lot of times we combine with insertional inactivation because the reporter genes basically let you visualize things. So these are things like color changes, like seeing blue color of colonies or white color of colonies. So this is the insertional inactivation method of trying to see if your bacteria successfully took in the insert or the new gene. Okay, this is basically what you see in the blue and white ligation transformation experiment. So you have this plasmid over here. Okay, in this example, it's not the blue and white case. In this example, the plasmid will have two different antibiotic resistance genes. Okay, this top one is antibiotic resistance for ampicillin, for instance. This bottom one is antibiotic resistance for kanamycin, for instance. Okay, so AMP resistance and CAN resistance. Now, if you successfully cut open this vector and insert a gene, well, what do you notice? That gene just destroyed the kanamycin resistance. Okay, so any bacteria that has a plasmid that simply religated on itself and never took in a gene, they'll still have AMP and kanamycin resistance. If, however, it took in the new gene, it'll have AMP still working, but it will not be kanamycin resistant. Okay, so in that case, what you would basically do is you would stamp the colonies that you got from this experiment on two different plates, because we have the ability to just stamp them, meaning you put a velvet cloth, you pick up your colonies from your transformation, and you gently touch it on an ampicillin plate, and you gently touch it on a kanamycin plate. And you look to see which of those colonies from your transformation plate successfully grow on ampicillin, but then die off in kanamycin. And you still have the original transformation plate to go back and take those colonies and grow them and use them for your experiment because they have the intended gene. Again, if you're having any trouble understanding this one, just send me a remind message and I'll draw out a picture for you to help you visualize this. Okay, now in the case of our lab experiment, what we do instead of having two antibiotic resistant genes, you have the idea that the vector has the first part of the beta-galactosidase gene, and then the bacterial strain has the second part. So when the regular vector meets the bacteria, boom, you get the blue colonies. Remember, we drew this out on the board. Now, if you then insert a gene into that vector, that beta-galactosidase gene is no longer functional, which means you can no longer get blue colonies. Instead, the white colonies are your success. Okay? Again, if you have any trouble, just send me a remind message and I'll explain further. Now this one here is basically what we were just talking about, just to give you a visual idea. This is the idea of the plasmid, so just like you did in your experiment, the plasmid having the first part of the beta-galactosidase gene, the bacterial chromosome having the second part. You put the plasmid into the bacteria, boom, you have active beta-galactosidase that produces blue colonies. If you then insert your gene right smack in the middle of here in the plasmid, well then you know that was successful when you no longer get beta-galactosidase working and you get the white colonies. Okay, so this we went over in lab as well, so this should be basically a review. Now, in addition to plasmid, sometimes we use some other types of vectors. Here we have bacteriophage lambda vectors. That's basically saying that you can use lambda, which is a virus, okay? A virus that infects bacteria with its viral genes. And interestingly, the viral genome of lambda is not actually all essential. So there's a portion of it that you're able to just, like you see in this figure, you're able to just cut out part of that genome and glue in whatever genes you want and put it back into the bacteriophage, into the virus. And now that virus will go and inject that DNA, including the gene you added, into bacteria. Okay, so this is just an alternate route that sometimes researchers use. And when you do this, when you have a viral vector introducing DNA into bacteria, we call that transduction. Okay, so in lab you perform transformation. This here is transduction. Now, another type of research vector that is commonly used is what we call the TI plasmid. This one's used for plant engineering. TI stands for tumor inducing, okay, not a famous musician. So it is tumor inducing, and the reason it's called that is because it's actually naturally carried by what's called Agrobacterium tumefaciens. And you have all probably seen this at one point or another. That's actually a type of bacteria that gets into plants and especially trees through cuts in their, in their bark. So we call that wounds. So for instance, if your parents ever yelled at you, if you're, basically carving into a tree or picking away, peeling with your finger pieces of the bark, when you wound that tree, you open it up to this bacteria. And that bacteria carries a plasmid that has what we call tDNA. And the tDNA, or tumor DNA, basically what that does is what it says, it's tumor causing. It integrates into the plant genome. And what that tDNA has is auxin genes, cytokinin genes, and opine genes. And these are genes that produce enzymes that, as you can see from down here in the figure, promote cell growth, cell division, and feed that bacteria. Now, when you think about this bacteria is putting genes that give enzymes to produce more cells, more plant cell division. What is that? That's cancer, right? That is plant cancer. And that's when you see those giant bulbous tumors sticking out of trees that you're going to see on the next slide. So anytime you're walking by or driving by any of the trees, even on campus, that have these big growths sticking out of them, I want you to shout Agrobacterium tumefaciens, okay? Just shout at it. Look at it. You see, look at that tumefaciens. And all your friends will, of course, think you are the coolest person ever because, that is awesomely cool, of course. Okay? Now, we can then use these plasmids in genetic engineering because if this bacteria has the plasmid, which mediates transfer of DNA segments into plant cells, well then what we can do is we can cut out those genes for the tumor induction and instead put our own genes in there. And the rest of that plasmid is very good at integrating into plant genomes. Okay, so we're able to modify that plasmid, the TI plasmid, to take out the bad bacterial genes and put in our desired genes, and boom, now we can get the plants to produce whatever we want. Okay, So to help you visualize what I was just talking about a minute ago, this is that, crown gall tumors that you might see on campus or various places when you're walking around or driving around. So remember to shout, look at that tumor fascians, okay? And this up here demonstrates what we were just talking about in terms of using the TI plasmid for genetic engineering. So you're able to cut it open, remove those tumor-inducing genes, and ligate in the genes of your desire. Then when you introduce this plasmid into plant cells, well, that plasmid, as you can see here, is naturally very good at integrating into integrating into plant cells and into plant DNA. So now the plants produced from that cell will have your desired genes. So for instance, those genes, think about what might you want to give to a plant. What kind of traits? Well, maybe it's a gene to produce bigger fruit. Maybe it's a gene to produce more fruit, more vegetables, okay, more product. Maybe it's a gene to grow taller, grow bigger. Okay, any of these things resist pests such as, bugs that might try and attack those plants. So that's how we do genetic engineering a lot of times when it comes to plants and crops, is we use the TI plasmid. Okay, make sure you know if you see a crown gall tumor though, that's thanks to Agrobacterium tumor fascians. Okay, remember that, it may pop up again. Now, when we talk about genetic engineering, all of this that we're talking about is being able to cut and manipulate DNA. So remember we said nucleases are the enzymes that we use to cut DNA. And there are exonucleases and endonucleases. So if you think of what does exo mean, well, that means outside, right? So exonucleases will cut at the end of a sequence, okay? Whereas endonucleases will cut in the center, okay, within the sequence. So now, with these exo and endonucleases, keep in mind earlier I said that each of these nucleases will recognize a particular sequence, which is usually a palindromic sequence. And it will only cut at that particular sequence. And each enzyme will have a different sequence a lot of times. And like I said, if that sequence is only once in the DNA, then that enzyme will only cut once. If it's 5 times in that DNA, the enzyme will cut every single time it sees that sequence. Now, there are 2 terms I want you to know the difference between. So circle, star, highlight these because these are 2 of my favorite words in the English language. We have isoschismers and we have neoschismers. And both of these are terms for nucleases, but how they differ, okay? The trick that I use to remember which is which, I think of neoschismers first, because whenever you hear neo means new, okay? So new means that neoschismers are 2 enzymes that will recognize the same sequence of DNA, but will cut at different spots. Okay, so they recognize the same palindrome, but will cut at different spots. For instance, one may cut here, so look over here, one may cut the sequence after the G, whereas the other enzyme, the neoschismer of that first one, will cut after the A or after that A. So they both recognize the same sequence, GAATTC, but they cut at 2 different places. Okay? Now, isoschismers, on the other hand, it's a little confusing when I say this. They recognize the same sequence and cut at the same spot. So isoschismers would both recognize GAATTC, and they would both cut after the G. Okay? That would be isoschismers. Now you may ask, well, if they're both recognizing the same sequence and they're both cutting at the same spot, why did they have two different names, such as enzyme A and enzyme B? That makes no sense. Well, it makes sense in that they were extracted from different organisms. So let's say you found enzyme A out here on the, soil surface, and you found enzyme B out in an organism in the ocean. Well, yes, they do the same thing, but you name them different things because they're found in different places. And it's so important to know isoschismer and neoschismer because, for instance, if you're ever in, medical usage or research field, you run out of a particular enzyme. Well, if you know the isoschismer or the neoschismer, you may have access to it, right? Without having to delay your work or run out and order something new. Okay, so make sure you know the difference between isoschismer and neoschismer. If you're having trouble, just contact me on Remind and I will give you examples. Again, I will draw them out if you would like. The other point that I want to make about nucleases is that they'll cut the DNA either leaving a sticky end or a blunt end. And you can see that in this top figure here. So if an enzyme cuts straight down and there's no overhang, as you can see here, that's a blunt end. Whereas if it cuts like this, cuts after that G, cuts after that G, well, now you have that little bit of overhang when they separate. And those are considered sticky ends because think about it. This T is all by itself now, and it's going to want to find an A, okay, to stick to. So that's why they're sticky, because they like to grab a hold of other single strands. And sticky ends tend to work the best for research because, think about it, it's sticky, it wants to bind, okay? The last thing that we have here is modifying enzymes. And modifying enzymes basically means that these enzymes can add a methyl group, which you see in this figure here, and that methyl group actually prevents restriction cuts. Okay, so if you have methyl groups present, it will prevent cutting of that DNA at that particular location. The last thing that we have here is ligases, which you already know that term. Ligases are basically like glue for DNA. They covalently link pieces of DNA, so they're very valuable to manipulating DNA because what good is cutting open that DNA if you don't have the glue to stick back certain pieces together? Okay. Again, if you have any questions about any of these terms we went over on this slide, just send me a remind message. Okay, so when we talk about genetic engineering, we also have what's called gene libraries and DNA libraries. Now, basically what this means is not only are we capable of cloning single or individual genes, but now we can clone libraries of whole genomes, okay? So as you see in this figure here, we can take an organism, let's say a bacteria's entire genome, so every gene it has, and what we do is we chop it up and each piece ends up in a plasmid. And then we do transformation and you get a single plate that has all of the genes of that bacteria's genome represented in that single plate. Okay, now keep in mind, each colony will not have every single gene. Okay, it's the whole plate that's the library. Okay, the plate will have all different bacteria, and each different colony will have different genes. So let's say this guy here has this first part. This guy here will have this second part. Okay, so each bacteria on that plate will have different genes, but the plate represents the whole genome. And since that plate represents the whole genome, you could then screen the plate to pick out whichever genes you want to work with. Now, there are also with gene libraries what we call metagenomic libraries. So they'll have a plate that has genes from many different bacteria. So a lot of times you see this, for instance, from an ocean environment. They'll collect a sample, and then they'll have a plate that represents all the genes in that particular location. They have prokaryotic gene libraries, which are, for instance, bacterial. They also have eukaryotic libraries. So these will have a lot of times yeast genes, or even you can do this with whichever organism you want. But basically the premise is the same, that the final plate will have the whole library represented. Then that brings you to the next point. Once you have this plate with all of the genes of an organism represented, well, what do you do with it? You have to screen it, right? Because now you want to know particular genes. Are certain genes there? Okay, where exactly in that plate are certain genes? So there are two techniques that we can use for screening a gene library. You can use hybridization, or you could use immunological screening. For hybridization, I want you to write down that this is for DNA. So you're using DNA probes, you're using single-stranded complements that are nice and sticky, and you're screening the library for the DNA, the actual genes. Immunological, you're now using antibodies to screen for gene products, okay, the proteins produced by the genes. Okay, so hybridization, you're going to be looking for DNA. Immunological, you'll look for proteins, particular proteins. Okay, so the first method is hybridization screening. Okay, I'm going to go through first, and then I'll give you the recap slide for you to take down your notes. So the first thing that you would do with hybridization screening is you would transfer your bacteria, which contain the fragments of the library, onto a filter. Okay, so transfer all of these bacteria from your plate that have all the genes present, and you transfer them onto a thin filter. You then lyse these bacteria, and you denature the DNA with alkaline or very basic solution. Okay? So now when you have denatured DNA, what do you know about single-stranded DNA? Well, single-stranded DNA will like to try and find its partner. Okay, it will want to stick to its partner. Okay, so what you do then, once you have this membrane of all the single-stranded DNA of this library, you then cover that filter with the solution that has radioactively labeled single-stranded DNA probes that complement the exact gene that you're looking for. Okay, you then develop that, and wherever you see the black, or in this case blue spots, that tells you that that's where your target gene is. Okay, that's how you find your target gene. So please remember that for hybridization screening, the key here is you take all of the bacteria of the library, you transfer it onto a membrane or filter, you break open those bacteria so it's just the DNA that you denature to make single-stranded, and then you target it with highly specific single-stranded probes that you can then detect. And those single-stranded probes will only bind to the target gene that you're looking for in that big library. Okay, so I'm going to give you a recap slide so that you can take down notes. Okay, so here you can pause the recording and write down the notes. The other way that you can screen a gene library is immunologically. meaning you use antibodies. So now it's the same kind of concept, but now looking for gene products, looking for particular proteins, because not only do you want to know the bacteria has the plasmid, but you want to know that it's functional. Okay, that you know which ones are actually producing the protein of interest. So what you do is, just like with the other one, you take those colonies from the plate, okay, and this time you're transferring to a membrane, and you're lysing them just like in the hybridization. So now you break open all the bacteria from the gene library, but now it's the proteins that you're targeting. So you use a different filter, a very particular membrane filter that the proteins will stick to. And the specific protein that you're looking for, you can then target with a primary antibody, because primary antibodies are specific to exact proteins that you would look for. You then apply the more general secondary antibody, which will bind to any protein, right? Any antibody. And that secondary antibody, what's important about that one is that it has detection on it. Okay, so it'll have some sort of probe or detection system on it. Usually we use chemiluminescence. So basically you want to see fluorescence. It's kind of like thinking in terms of a Western blot, where you get the fluorescent detection. So kind of think of this like screening for a particular protein, where it's almost like hybridization, but now instead of DNA, you're looking for a protein product. You take the gene library, you crack open all of those bacteria onto a membrane where their proteins will stick, and then you look for the exact protein that you want. And keep in mind, through this, whether it's hybridization or immunological, you still have the original plate of bacteria. It's kind of like you made a copy of it, right? So once you find the location with the fluorescent detection, you know exactly which colony to go grab and allow to grow. Now, when we talk about biotechnology and genetic engineering, one of the most powerful techniques that you'll hear about is polymerase chain reaction, or PCR, which is a way to amplify DNA. Okay, so here we have PCR, which, like I just said, is polymerase chain reaction. Its purpose is to amplify DNA, and basically it makes a ton of copies of a very specific piece of DNA, whichever piece of DNA you're targeting. And you may ask yourself, well, why would you want to do that? What scenarios? Well, it's important in things like crime scene investigation, paternity testing, evolution, diagnostic medicine. So you use it a lot in hospital labs as well. Now, in order to perform a PCR, there are various things you need. The first one is the DNA template. So basically the target DNA that you want to amplify. Okay, you need forward and reverse primers, which are little pieces of single-stranded DNA that match the beginning and the end of the DNA sequence that you want to target. Okay, because as you know, polymerases can't start from scratch. They need something to have as starting points. Okay, you'll have the DNA polymerase itself, which is the enzyme that will build the copies of DNA. And it has to be temperature stable, because during this process, you're going to, be using a machine that goes to high temperatures to denature the DNA, so the enzyme has to survive that. You need what are called dNTPs. Those are your nucleotides. So picture those as your A's, your T's, your G's, your C's, the bases that make up DNA. You then need buffers and magnesium chloride, because think about it, you need cofactors for that polymerase. The DNA polymerase is an enzyme, so you need cofactors to help it work, to activate it, and you need to maintain a good environment for this whole process. And the piece that students always forget, you need what's called a PCR machine. You need a very particular machine that will cycle through. It'll go to high temperature, cool down. It'll fluctuate temperatures for you for this process. Okay? Now, with that fluctuating temperatures, the reason that is that each PCR, there are 3 steps to happen. You need the machine to first denature the double-stranded DNA, which happens at high temperatures. So you see that here. You heat, the machine will go to a high temperature to separate the double strands of DNA. It will then cool down so that the short primers targeting your specific sequence can bind. Because think about it, if the machine did not cool down, what would happen? These guys would denature too. They wouldn't be able to stick. So it goes from hot to cool, and then it goes to the temperature for Taq polymerase or whichever DNA polymerase you use, because each enzyme has an optimal temperature. Okay, so the first step is high temperature to separate the double strands so that you can then cool the temperature to allow the primers to stick, and then go to the optimal DNA polymerase temperature, such as 72 degrees Celsius, for the polymerase to then build from that primer, okay? And it makes copies of that DNA sequence. Now, you don't have to be able to draw all of this out, but this is just to show you that you don't get your target DNA until 3 cycles later, because each time those primers are, building the polymerase is building from the primers, you end up with a lot of overhang sequences. And as you can see here, this is when you get the nice target DNA. Okay, so you don't have to be able to draw this out or anything. Just remember the fact that it's not until 3 cycles of PCR that you finally get the properly sized target. Okay, circle, star, highlight, 3 cycles. Now, when we talk about PCR, there's a lot of different types of PCR that, basically they have made this technique more and more powerful for us researchers. One of the ones that we use a lot is called qPCR, or quantitative real-time PCR with TaqMan probes. Okay, what this means is not only can we amplify or make a ton of copies of the DNA, but we can quantify it as well. So this one will quantify while it amplifies, because it's using these special TaqMan probes. And what these TaqMan probes are, you have to know what they are. They have a fluorophore on one side, so a fluorescent probe on one side, and then what's called a quencher on the other side. A quencher, like when you quench your thirst, it blocks the fluorophore, okay, because right now it's connected to that guy. So when the quencher is near the fluorophore on this TaqMan probe, the quencher blocks fluorescence. What then happens is this TaqMan probe, it's a single-stranded piece of DNA that binds in the PCR to the target DNA that's getting amplified. What happens when the polymerase is amplifying that DNA? Taq polymerases have what's called exonuclease abilities. So that Taq polymerase, as it's building the new strand, it will then come along. Once it makes it to this probe, it'll chop it up. So when the Taq polymerase here chops up that probe as it's building the copy of DNA, what happens? The fluorophore is now floating away. It's separate from the quencher. What happens when the fluorophore is separate from the quencher? It fluoresces. Okay? So the more initial product that you have, the more template DNA you have, the more new strands will be built, the more probes will get cleaved, and the more signal you'll see. So that allows us to quantify it, okay? So one more time, the more product you have, the more template DNA you have, the more new strands will be built with the Taq polymerase, which means more probes get cleaved, which means greater fluorescence as the fluorophore is separated from the quencher. Okay, this is a slide that helps you kind of see in words what I was just saying. Make sure you're very comfortable with knowing what happens with the fluorophore when it's near the quencher versus when it's separate, okay? Make sure you can explain how exactly does a TaqMan probe allow us to quantify a PCR product, okay? Similar to that, sometimes you will hear reverse transcriptase PCR. What reverse transcriptase PCR is now you're amplifying genes using mRNA as the starting point, because as you know, reverse transcriptase means you're going from RNA to DNA. Now, why would you want to use mRNA as a starting point of your PCR? Well, this is used in gene expression research. Okay, because mRNA is gene product, so it shows how active particular genes are. Okay, so you're now going to have to clone copies of intron-free DNA in this process and amplify that. So the main steps of this, of a reverse transcriptase PCR, first you have to isolate that RNA, the mRNA. You then clean it up to get rid of any DNA that might be contaminating it. And then you do what's called first-strand cDNA synthesis. And you do not have to memorize these steps of first-strand synthesis. It's basically meaning that the machine will make the complementary DNA copy of that RNA. Okay? And then once you have the DNA copy of that RNA, now you can put it into the PCR and amplify it, okay? Because PCR only works for DNA. PCR can only amplify DNA. So if you want to study RNA with PCR, you first have to make a DNA copy. So that's why you need reverse transcriptase PCR, okay? Keep that in mind, circle, write down that note, put little stars next to it. PCR only amplifies DNA, okay? Now that reverse transcriptase PCR is very valuable to us in the research field because it allows us, like I said, to examine gene expression, okay? So you can look at whether a gene's expression is upregulated or downregulated in certain conditions, basically meaning you'll have two different conditions and you'll ask yourself, when do I see a greater signal? When do I have more of that fluorescent final product? So if you see a lot of reverse transcriptase PCR product, well, that means that there was a lot of mRNA present to turn into the DNA and make copies of it. Right? So that means that gene expression was high in that condition. Whereas if you do not see much fluorescence or amplification at the end of the reverse transcriptase PCR, well, that tells you there wasn't DNA to copy or amplify because there wasn't mRNA to copy, which means the gene was not being expressed. Okay, so reverse transcriptase PCR lets us look at how much gene expression there is in different conditions. Okay, so it's a very powerful technique. There are also a lot of medical applications of PCR, whether you're doing regular PCR or the big time qPCRs. Okay, and with these, bullet points that I have on this slide, keep in mind that there's a whole lot more you can do with PCR. This is just the sampling. It's very powerful technique because you're able to amplify or make copies of very small amounts of DNA, which if you think about it, well, that's great in forensics, especially since at a lot of crime scenes, you're not getting a lot of DNA or it's been highly contaminated. So this allows you to kind of amplify your sample to work with. It's also very powerful in diagnostics because we know the genomes of almost every bacteria or virus that are most commonly infecting people. So whenever we find new viruses, new bacteria, the first thing we want to try and do is, get its genome, sequence its genome. And then once you can do that, you can use PCR to see does a patient have it or not. For instance, you use primers that target sequences found in that virus or that bacteria, and you test your patient's blood samples or swabs or pus samples, anything like that, just to see, okay, if this person's infected, the PCR primers will pick up the viral or pathogen genes and make copies of it. And you'll see those copies on a DNA gel, like on the bottom of this slide here. Okay, that's DNA gel. You can see bands when the amplification occurred. Now, if that patient's not infected, the primers won't find anything. They won't have anything to bind to. There won't be any amplification, and you won't see any band on the final gel. Okay, and they do this for a lot of different infections, and they even look at mutated genes like BRCA1 for breast cancer. They'll have primers that target the mutated sequence and see if that's present in a sample. If you see amplification, that means the gene you're looking for is present in that person. And if you don't see amplification, then the primers had nothing to stick to. There was no bad gene present. Okay, so very powerful in the medical field. It's also very powerful in the environmental field. Okay, environmental field. I'm not going to go into too much detail here, but I basically wrote out a lot on this slide just in case you're more curious about how it's actually used environmentally. But basically, you're able to target bacterial genes to see if they're contaminating certain areas, or to find particular metabolic aspects that might be valuable from different bacteria. So for instance, if there's a hot spring, these very crazy conditions that can withstand heat, well, let's gather the microbes there and amplify their genes, see if there's a product that we can use. Okay, so we're able to find very valuable enzymes for our research, even things like the PCR polymerases that we use. We have gotten from, trawling ecological samples, looking for amplified gene sequences. And then the last one here that we have is the idea of rescuing DNA from extinct life forms with PCR. Now, this is kind of like the Jurassic Park type of scenario. It's not completely, realistic in some senses, because cloning such large amount of genomes can get problematic. A lot of times the vectors can't handle it, or you have a lot of mutation issues or instability issues. But just be aware that trying to rescue old forms of DNA is one other possible use of PCR, because basically once you look at sequences, you have sequences. If you can make little primers for them, you could amplify or target any particular genes you want. Okay, so we're not going to go into crazy detail. I just want you to know that there are environmental and extinct life form uses for PCR. The last few slides I want to cover are the molecular techniques for analyzing DNA. So with blotting, there are a few different forms of blotting that I want you to know the difference between. Now, in all of them, you'll have fragments run on some sort of a gel, then copied onto a porous membrane, and then probed to have some sort of detection. Now, what I want you to know is the difference among southern blot, northern blot, and western blot. And some students like to use the trick of snowdrop to remember that southern blot is for probing or detecting DNA, northern blot is for detecting RNA, and western blot is for looking at proteins. Okay, so in all of these, you're putting fragments onto a membrane, but in southern blot, you're probing for DNA. In northern blot, you're probing for RNA. In western blot, you're probing for proteins. Okay, and it's more specific blotting than simply running a gel electrophoresis. Now, gel electrophoresis is one of the most commonly used techniques. What you basically do is you put DNA fragments into an agarose gel, and that agarose has a network of varying size pores. You put in a salt buffer, and if you think about it, salt conducts electric currents. So you apply an electric current, and as you already know, DNA has a negative charge. So when you put that electric current through, those negative charges are going to flow toward the positive side of that tray that you have set up. Okay. Now, in addition to your DNA samples, you also load a marker or a ladder, and that's important because it will have known size fragments of DNA that you can then compare your sample to and figure out exactly what size DNA you have in your sample. Now, with this concept, you have the negative DNA moving its way, navigating, snaking through the pores of that agarose gel, trying to make it to the positive side of that gel. And be careful, because it is possible for your DNA to flow right through, it's so desperate to get to the positive electrode that if you leave it too long running, your DNA can run right off of the gel into the salt buffer. Now, the way this gel concept works is smaller and larger fragments will move slightly differently. So think about it. These DNA fragments, they have to navigate, snake their way through the pores of this gel. If they're bigger fragments, it's kind of like when you see adults in a children's playground and they're running after their child. And sometimes the adult, because they're big, the playground's not meant for their size, they'll get stuck, trying to navigate through. How many times have you seen on YouTube, some big parent getting stuck on a slide or something, those tube-like slides? So that's what happens with larger fragments. They get stuck early on in the gel. Okay, so they'll stay very close to where you loaded them into those wells. Whereas the smaller ones, they can zigzag just like little kids running through the pores, and they make it farther. Okay, so larger fragments are closer to the wells where you put them, and smaller fragments of DNA on a gel will run further. The last important thing when you're doing gel electrophoresis is to remember to have ethidium bromide. And ethidium bromide has two functions in gel electrophoresis that I want you to know. One, it's what we call an intercalating agent, intercalating, intercalating agent. That means that it pulls between or collects between the base pairs of DNA. The second characteristic is that it glows under UV light. So when you put these characteristics together, it means that when you have ethidium bromide in your gel, it will only end up located where the DNA is because it pools or collects in the DNA bases. Then when you shine UV light on this gel, it'll fluoresce, it'll glow for you. So it'll show you exactly where the DNA is. So this is just a visual to help you see. Again, you load your DNA into wells on the negative side of the tray, and then these smaller fragments will run further. They'll be further away, trying to make it to the positive side, whereas the ones closer to the wells will be larger sizes of DNA. And then you use UV to visualize. And why does that allow you to visualize? Because of ethidium bromide. Now, in addition to visualizing DNA in terms of the gels, you're also, when you do research, able to figure out the concentrations of your DNA and RNA. And what's really important here is because in a lot of our experiments, when we try and isolate DNA or RNA, a lot of times the samples can get contaminated. And it's, it's natural. There's so many proteins everywhere around you that when you're doing these experiments, you can be very careful, and still your sample, even from the cells that you're cracking open, sometimes you try and clean up everything, but things happen. And so you're able to use UV spec or UV spectrophotometry. Sorry, I mess this up every time, even though I've used it for over 15 years now. But yeah, spectrophotometers or spectrophotometry, that's why I just say UV spec. You can use UV spec to kind of see how pure is your sample. And I want you to be able to tell me, basically in questions like on an exam, to be able to tell me if I give you what's called a UV spec ratio, 260 over 280, you should be able to tell me how pure of a sample, whether it's pure DNA, whether it's RNA contaminated or protein. So basically the concept is DNA and RNA absorb UV light at 260 nanometers. Okay, so think of 260 as DNA and RNA, whereas 280 on a UV spec represents proteins. So when you then do the ratio of 260 over 280, pure DNA will have a ratio of 1.8. Circle, star, highlight that number. 1.8 on a UV 260 over 280 tells you you have pure DNA in your sample. You did a beautiful job of isolating DNA. If protein is present, well, what did we say protein is? 280, which is the bottom of our fraction. So if protein's present, you just increased the bottom of a fraction. So for instance, you now went from 1 over 2, 1/2, to 1 over 4. What do you know about 1 over 2 versus 1 over 4 now? Well, 1 over 4 is a smaller number, right? The bigger the bottom of that fraction gets, the smaller your ratio is. So if you end up with a UV 260 over 280 that is less than 1.8, you have a lot of protein present. So you'll usually see it as like a 1.3. So if I ask you, what does it mean if your DNA sample has a 260 over 280 ratio of 1.3, well, it tells you that there's a lot of protein contamination. Okay, make sure to write that down. If RNA is what's actually contaminating your sample, then instead, this top number gets bigger, okay, because RNA, the difference in terms of uracil and the single-stranded nature, it ends up having the pure ratio of 2.0. So if you see 1.8, your sample is good and pure DNA. If you see something like 2.0 or 1.9, you have RNA contamination. If you see something like 1.6 or 1.3, you have protein contamination. Okay, make sure you're comfortable with those 3 scenarios. If you're unsure about this, just send me a message in the Remind app and I'll give you the exact format of how to practice questions like this. The last thing that I want to do here is just to briefly mention the idea of applications of biotechnology and the kind of ethics that you encounter, okay? Because it's a big thing today, you know, especially with the media kind of blowing things out of proportion and social media existing. Biotechnology gets a lot of bad rap, and it's mainly because people don't necessarily understand the science behind it. Now, biotechnology, the exact definition is when you're using living organisms to create a product or to kind of modify products to help humans or other organisms, okay? So the idea behind it is to be helpful, but a lot of people are scared about it. Genetically engineering organisms has been used a lot of different ways, biologically, pharmaceutically, producing things like insulin, which is very valuable to people. When you do these things, such as you kind of see on the bottom here, genetically engineering corn to what we know it today, fish to increase size, for instance, or increase the amount of fruit, or to even help produce things like, in this case, immune response. So rather than having old school vaccines, but giving third world countries fruit that trigger immune response. All of these things, they're meant to kind of use genetics and biotechnology to help people. It's using what we talked about in this lecture to introduce new genes, which introduce new proteins. Okay, so once you do this, those organisms are now called transgenic because they have new genes put into them. It's used with things like vaccination, crops, medical, like I said, things like insulin. Okay, a lot of times the things that we're trying to do with genetic engineering, especially in agriculture, it was to make crops more robust. So produce more fruit, like I said earlier, produce bigger fruits and vegetables, have healthier plants, bigger size, resistant to things. They do this with animals to make them bigger, more meat, more, taste, enhance the products. And like I said, medically, things like, producing increased amounts of insulin or various hormones or gene products that we need. Okay, so you can kind of look into it more, make your own decision whether you're for or against, but the way I see it is it's done a lot of good. Now, there are concerns from people because they think that it's too new to kind of see the full implications, and they worry that there may be bad side effects down the line. But at this point, we have had quite a few generations to look at these things. But, time will tell. So always research, look into things from reputable sources, truly vetted, peer-reviewed journal articles, rather than listening to the media. Don't let social media, don't let news media make the decisions for you, okay? Because even as you see with things like coronavirus or E. coli outbreaks, if you don't know the science and you don't look it up, you can start believing the scare tactics and the false reports that they make about things like those events or vaccines or GMOs. But be your own advocate and really look into these things, because you'll see that a lot of the so-called bad things that they try and say about things of science, not true. Okay? So this is just a touch upon the subject. If you're more interested, make sure to look more into biotechnology, because it's a huge field now. It has a lot of potential value. Okay, so that is it for today's lecture. Keep in mind that the exam is on lectures 3, 4, and 5. So you now have everything you need to study and prepare for the next exam. Make sure to head over to the discussion board and post your 2 questions. Take a look at everyone else's questions, because some of them may pop up again. And as always, make sure to send me a remind message for any of the slides or information that you would like more information or practice problems on. Okay? I have no life. Never hesitate to ask for help. Okay?